Welcome back to my channel, Back and by Wall. In today's video, I'll be discussing the differences between prokaryotic cell and a eukaryotic cell, and I'll be discussing it by a diagrammatic way as well. And first is a table, and in this table there are components, there are various components, and we will be talking these components in with respect to the prokaryotes and the eukaryotes. Okay, first comes the nucleus. A nucleus is a well confined, we can say, a compartment which consists of the genetic material. This confined compartment is only present in case of the eukaryotes and it is absent in the prokaryotes. The genetic material in case of the prokaryotes, it is not termed as nucleus, it is termed as nucleoid. Okay, and it is in direct contact with the cytoplasm, where in case of the eukaryotes, the genetic material is confined within a double layered membrane and it is separated from the cytoplasm, whereas it is in contact with the cytoplasm through the nuclear pores. Okay, next comes the diameter. And the prokaryotic cell, generally if I talk about prokaryote, that is bacterium, that is E. coli, which you will be familiar about, the diameter, it is 1 to 10 micrometer, and it can vary among the prokaryote. This is just a general data, and in case of the eukaryotes, it is about approximately from 10 to 100 micrometer. Okay, the various cells which are found on the eukaryotes in the plants and the animals, the size of the cell can vary between 10 to 100 micrometer. Next are the membrane bound organelles. Okay, in a cell, to perform a function, that is we can say, to keep it viable, to keep it living cell, it has to perform a number of functions. These various different functions, they are usually performed by the organelles. Okay, and membrane bound organelles are usually only present in case of the eukaryotes, whereas in case of the prokaryotes, they are some of the organelles, or we can say only kind of, we can say, ribosomes are present, and these are usually not membrane bound. Okay, and if I talk about ribosome, I have just given you an example. Okay, that only kind of organelle which is ribosome, that is the protein manufacturing unit, we can say. Okay, and in case of the prokaryotes, it is usually 70s. Okay, whereas in case of the eukaryotes, it is usually 80s, which is usually found in the eukaryotes. And okay, 70s is usually, I think it is found in the chloroplast and the mitochondria. Okay, next come the genome, that is a genetic material. Okay, genetic material, we can say, it is a main part, okay, which controls, or which we can say gives a command to a cell what to do, how to do. It has the code within it. Okay, in case of the prokaryotes, it is just a single DNA molecule. Okay, and usually they are circular in kind of, we can say, nature. It will be more clear in the coming slides. And next, in eukaryotes, the genetic material is present in the form of chromosomes. Okay, and for the movement, prokaryotes are having a flagella. Okay, they have flagella for motility and I can also say to grasp their prey. Okay, in some organisms, flagella is used in that case as well, whereas in eukaryotes we do not find any kind such of, we can say a part of the cell. Okay, there is no flagella in case of the eukaryotic cell in the plant or the animal cell. The constituent of, we can say, the main part which is forming the cell wall, we can say the compartment which separates it from the environment, is peptidoglycan in case of the prokaryotes. Okay, whereas in case of the eukaryotes, it can be cellulose, chitin, pectin. Okay, and next, the size of the prokaryotes are usually smaller. I have already told you the diameter, that is of prokaryotes is in case of prokaryotes 1 to 10 micrometer, whereas in case of the eukaryotes it's large. And the division of the prokaryotes takes place with the phenomena which is termed as binary fission. Okay, that is, fission is the separation, we can say, the dividing, and binary means two, that is the parent cell divides into two. It will be more clear in the coming slides. And whereas in the eukaryotes, the cell division takes place in the mitosis, that is somatic cell differentiation takes place by mitosis, whereas the germ cells which is found in the animal, in case of the animally, we can say, kingdom, it is usually with the meiosis as well. This in the coming slides I'll be discussing some of the, we can say, the parts of the both differences in the eukaryotic and prokaryotes with the help of diagrams. And as you can see in this diagram, I have tried to make it clear the genome, that is the nucleus. We can say, I told you that in case of the eukaryotic cell, the genetic material is usually confined by a double layer membrane, and it is separated from the cytoplasm with this double layer membrane, and it keeps a contact with the cytoplasm with the help of the nuclear pores. In case of the prokaryotic, the yellow part, okay, the yellow part is the nucleoid. This is my nucleus. This is, we can say, the prokaryotic nucleus. Okay, and there is no, you can just see from the diagram, there is no compartment. Okay, there is no compartment, and it is just randomly like this coil. It is circular genome. Okay, and lights, it is in direct contact with the cytoplasm, and this thing is plasmid. Okay, this is a plasmid. This is a plasmid. Excuse me for the handwriting. And this is a plasmid, and basically what are plasmid? These are the extra chromosomal genetic material. Some of the, we can say, the prokaryotes, they have extra chromosomal genetic material, and they're usually termed as plasmid, and they can play a number of different roles, like we can say antibiotic resistance. Okay, and a number of other roles are also played by these extra chromosomal genetic material. Okay. Next comes the size of the cell. The prokaryotic cell is usually 1 to 10 micrometer, whereas here in the eukaryotic cell, this is a plant cell, and the size of the cell ranges from 10 to 100 micrometer. Okay. Next comes the membrane bound organelles. In case of the prokaryotic cell, these are absent. No organelles are present. Membrane bound organelles are absent, whereas some of the membrane less organelles like ribosomes are present. Whereas in case the eukaryotic cell, that is we can say the two kingdoms, animae and plantae, there are number of membrane bound organelles. The mitochondria, we can say the powerhouse of the cell, it generates the ATP for us to function, for the cell to function. Next is the Golgi apparatus, where the packaging of the, we can say, the proteins which are synthesized from the cell, the packaging, the modification takes place in the Golgi apparatus. And this here is chloroplast. Okay, we can say the kitchen of the cell, where the this is usually found only in the plant kingdom. It is not found in the animal cells, and this is a site of the photosynthesis, that is the synthesis of the starch. Next, here lies the lysosome, that is the suicidal bags, and this is the RER, that is rough endoplasmic reticulum. This one is, the purple ones are the rough endoplasmic reticulum, whereas these are the smooth endoplasmic reticulum. Usually this rough endoplasmic reticulum, they are tightly bound with the nucleus. These are the invaginations of the nucleus as well, and whatever proteins, the proteins synthesized in the RER, this is the site at which the ribosomes are bound and the protein synthesis takes place. Okay, and that was all about the membrane bound organelles. And next comes the genetic material, and in case of the prokaryotic cell, it is just the chromosomes lies in contact with the cytoplasm. There is no confined, we can say, nuclear membrane, and there are number of other extra chromosomal genetic material, and the number of the extra chromosomal genetic material, that is the plasmids, can vary from cell to cell, from type to type of the prokaryotic cell. Whereas in case of the eukaryotic cell, there is a very, we can say, very compaction bound genetic material in the boundary, in the nucleus. This is my cell, this is my nucleus, this all the genetic material is usually packaged from, we can say, different level of organizations, and which in turn helps in the packaging of the genetic material. And because it's large, it's in meters, and to confine such long DNA, a long stretch of DNA into a micrometer, cell organelle is very difficult. So we need that level of cellular organization, genome compaction, and which leads to the packaging of that into the nucleus. Okay, these are the chromosome. This is the chromosomes. These are different levels of the genome level organization packaging. This is the DNA. DNA is bound to the nucleosomes. The nucleosomes are then further packaged into the, we can say, the scaffolds. These are the scaffolds, and a number of level of organization is there in case of the eukaryotic cell, and it is, we can say, far complex than the prokaryotic cell. Okay, I hope it's clear. Next comes the flagella. I already told you, only the prokaryotic cell. These are only found in the prokaryotic cell. These are usually used for the motility. Major function of the flagella is for motility. And this is the bacteria. And this is the purple shaded is are the flagella. Okay. Next is the multiplication. That is in case of the prokaryotes. I have already told you binary fission takes place. Fission means splitting. Okay, splitting of what? Splitting of my parent cell. Binary means two, that is into the two daughter identical daughter cells. This is a we can say a diagrammatic representation of the binary fission in case of the prokaryotes. In case of bacteria, first the parent cell, the genomic material, genetic material replicates. As after replication, it gets segregation along with segregation of genetic material, the replicated DNA. Septum is formed, and as at the end of what happens, there is a splitting of my parent cell into two identical daughter cells. Okay. Next is the mitosis in case of the eukaryotes, that is plant, and in case of the animals, that is interphase, my resting stage. The mitosis first stage, prophase, metaphase, anaphase, telophase. Mitosis is completed. Okay, that was all about the differences in prokaryotes and eukaryotes. Thank you so much for watching my channel. Please like, subscribe and share.